Okay, so the microscopy lab is just a lab to help us understand how the microscope works and gain some experience using it, okay? Because not everybody's used a microscope very much by the time they get to grade 10. Okay? So for our report, okay, same things apply as they always do. You need to put the problem in your own words in the form of a question. Okay, so you know, something about how a microscope works or how it would allow you to see small things, something like that, okay, in your own words. For our design, okay, the experiment is going to go like this. You are going to be looking at a letter E. You're going to make a wet mount of the letter E. You're going to look at an amoeba. You're going to look at a paramecium. And you're going to look at a euglena. Okay? So you have 4 specimens, 4 different specimens that you are going to place under the microscope. Okay? What we're looking for with those specimens is their appearance and the properties of what we see through the microscope. Because again, this lab is about how the microscope works. Okay? So we're looking at 4 different things, but we're going to make similar observations about all 4 of them. Okay? We want to see, what do they look like? Okay? Are they enlarged? Do they get flipped over? Do they change color? Right? All that kind of stuff. Okay? We're looking at the properties of the image that the microscope produces regardless of what we're looking at. Okay? We're going to make the same kind of observations. And in fact, we're going to make a lab diagram of each thing that we look at. Okay? I'm going to show you how to do that today. That's probably going to be the longest part of what we do today. And then in terms of the experiment, things that we want to kind of make sure are kept the same would be that, we would try to use the same microscope throughout the whole lab so that, nothing about the properties of what we're doing would change. Okay? All of our, slides are made of glass and things like that. Think of anything that, would stay the same each time you look at a different specimen under the microscope. Okay, and why that might be important, okay? Especially considering most important observations we're making are the appearance of the things we look at. Okay? So remember, each variable needs to be explained, okay? Identified and explained. One manipulated variable, okay? There's only one, and I kind of gave it to you already. Okay? There's only one responding variable. Okay? And I talked about that. Okay? Responding variables, always the things you're looking for or recording or making observations about. Okay? And then 3 controlled variables, each one explained independently. Okay? Okay, in terms of our hypothesis, okay, what are the 3 words that have to go in a hypothesis? If, and, then, okay? So the if part talks about what the premise is, okay? In this case, we're trying to understand how the microscope works. So the if part of our hypothesis should talk about how a microscope works. Did I show you that the other day? I kind of drew an ugly picture and showed the lines. That's how a microscope works. You might want to refer back to that. Okay, the part to put together the if part of the hypothesis, you know, about lenses and light and what it does to the, what the lenses do to the light. Okay, that should go in the if part. Okay, and part, as always, describes the experiment. Okay, so you're telling me, okay, in this experiment, 4 specimens are going to be observed under the microscope, and lab diagrams will be made of all 4. Okay. Their properties compared. The then part needs to predict the properties of your images. Okay, and that doesn't have to be terribly complex. Okay, I mean, the whole purpose of a microscope is to make things look bigger. That can be part of the then part, okay? Hopefully your microscope makes everything look bigger, okay? But it might also do a couple of other things to the images that you see, okay? And those would be important to predict as well. Or if you don't predict them, mention them in the conclusion after they happen. Okay? All right, give you guys a couple minutes to work on the variables there and the hypothesis. I'm just gonna grab a microscope real quick. Okay, so what we're going to be doing, okay, is looking at the 4 things that are listed here. The letter E, the paramecium, the euglena, and the amoeba. Okay, but we're not going to do them in that order. Right? We're going to look at the letter E first. We're going to look at the amoeba second, the paramecium third, and the euglena last. Because we're going to go from largest to smallest. As we gain experience, then we'll get better and we'll be able to find and focus smaller things better. Okay? Now, looking at the letter E is the start, because the letter E is big. Okay? So we're just going to take a cutting from a newspaper, okay, like from an article, not from a headline. So a lowercase letter E. And we're going to make a wet mount of that. Okay? How many people have ever made a wet mount before? Okay. All right. So to make a wet mount, what you do okay, so in the end, our wet mount is going to end up looking like this. Okay, the big rectangle here, that's the slide, a rectangular piece of glass. Okay, on there is going to be a letter E cut out of an article, just a letter E, okay, and placed on the microscope slide. Okay, then you're going to put a drop of water, that's the blue circle, and then you're going to put a cover slip, which is a small, transparent, square piece of plastic. Okay, so the way you do that is this. So this is the slide. Okay, I'm going to put the letter E, when it's cut out, on the slide, so that would be the newspaper. So now I'm looking at it from this direction, right? This is from above, this is from the side. Okay, then I'll drop the drop of water on there. And it'll make a bubble because water is what kind of molecule? Polar molecule. That's going to be important for the analysis, by the way. Okay, and then I'm going to drop the cover slip, okay, on like this. I'm going to set the cover slip at an angle on the slide, and then I'm just going to let this end go. It's going to fall down, and it's going to squish the water, okay, out to the sides, and the cover slip will stick because water's polar. Okay. Now, now you've made a wet mount. Pretty straightforward. It's really easy. Okay. The only thing we got to make sure of when we do this is that the letter E is right side up when we put it in the microscope. Okay, so I look at it. Okay, it looks like a letter E. I put it in that way because this is the only thing that we're going to look at whose orientation we can be sure of. I don't know what an amoeba looks like if it's backwards, upside down, or whatever. Okay, but I know a letter E. Right? When I look through the microscope, spoiler alert, the letter E is not going to look like this. Okay? As the light goes through the lenses, something happens to it. Okay? And we're going to look at it that way. Everybody with me on that? So that's the first thing we'll look at. And the letter E, you're only going to look at on the scanning power lens. So all of our microscopes, whether they're the new ones, that's the smaller versions, okay, or the old ones, the bigger ones, okay, have the same lenses, same magnifications. Okay? The scanning power lens magnifies a total of 40 times. Okay? So we're going to look at our letter E through the scanning power lens. If you go any higher than that, you won't see all of it because it's so big. All right, everyone with me there? Okay, and what you'll do then, once you've got it focused, is you will take a picture of it with your phone. Okay, and we're going to use that to make our lab diagrams. Be thankful you have that technology. Back in the day, you had to draw it by hand with a pencil. And I suck at drawing. Okay, it was the bane of my existence to draw lab diagrams back in the day. Okay, so be thankful you guys have the digital camera technology that makes life easy. Okay, so we'll just take a picture of it and then we'll label that diagram. Then you're done with that. Okay, you'll take the cover slip and the letter E and you'll throw them in the garbage and you'll wipe off the glass cover slip, or glass slide, sorry, and put it back in the container. Okay, then you'll grab the slide for the amoeba. Right? The amoeba is usually a pinkish purple, sometimes blue blob. And by blob, I mean not every amoeba looks like that. They generally are a blob, okay, that is generally that color. Okay? You'll know it when you see it, okay? It's going to be stained either pink or purple or blue, okay? And you'll know when you're looking at it, okay? They're fairly large. In fact, we'll look at them on scanning power, but we'll take our picture for our diagram on the low power lens, which is yellow in color on both, okay? So it's the yellow on the middle length lens. It magnifies 100 times, okay? If we go any bigger than that, you won't see the whole amoeba, okay? And we want to see the whole amoeba, so low power for the amoeba. And then you'll take the amoeba slide out, put it back in the container, and you'll grab the paramecium. The paramecium looks like this, okay? Kind of an oval shape. And if you get them really well focused on high power for these ones, because they're smaller, okay, you might even see these little hair-like extensions that come out of the outside called cilia. Okay, if you get those, you did an excellent job of focusing your paramecium. Okay, take a picture of that on high power, and then put that slide back. Then you will grab the slide for the euglena. This picture does not show the euglena as it's going to appear for you. It is about the same shape as the paramecium. But it'll look about like that, even on high power. Okay, they're not very big. Okay, in fact, when you first try to find them on scanning power, if you see little tiny blue slivers, that's them. There's going to be a lot of them, but they're really, really small. Sometimes it's hard to really actually see them on low power, or even, I'm sorry, on scanning power. You see them on low power, okay, then you'll see them again on high power. But they are very, very, very small. Okay, and so you'll take a picture of them on high power as well, and then you're done with the microscope. Okay, then we'll do our lab diagrams and move on to the analysis from there. Okay, everybody good with that? All right. Then let's talk a little bit about how we do a lab diagram. So this you're going to need to write down because it isn't in your notes anywhere. Okay, so the three lenses we use okay, have different magnifications. They also, as a result, have different fields of view. Okay, so our scanning power lens has the smallest magnification, but the greatest field of view. Okay, when I look through the microscope, it doesn't matter which lens I look through, I see the same brightly lit circle. Okay, but how big that brightly lit circle is different depending on what the magnification is. Okay, or how much area I see through that circle. It doesn't look any different to me, but the area of the slide I'm seeing in it changes and gets smaller and smaller as I increase the magnification. Okay, it's kind of like this. I'm at a Flames game and I'm trying to find my friend in the crowd. Okay, so my friend, like me, is an Oilers fan. So it's not going to be hard to spot because it's going to be the other guy in the crowd wearing an Oilers jersey. Okay? So I start scanning the crowd for an Oilers jersey. Okay? Now, I look around, I see lots of red, I see lots of red, then I see blue. I'm like, okay, that could be him, but he's on the other side of the stadium. So I pull out like a pair of binoculars. Okay? Now, when I was just looking for him, I could see that whole side of the stadium. As soon as I take out the binoculars, how much of the stadium do I see? A much smaller part. But I see it in much greater detail because it's magnified. Okay? And then I'm looking at it, I'm like, okay, that's definitely an Oilers jersey, but I can't tell the face. So I pull out a big telescope that's even greater in magnification. And I look through that. And now all I can see is like the whites of his eyes because I've like blown it up so incredibly large. I'm like, that's totally it. Okay? So that's what you're doing with a microscope. The scanning power lens allows you to scan a very large area of the slide at a low magnification and find what you're looking for. It's not generally used to discover anything or see a lot of detail. It's just for location. Okay? Once I find what I'm looking for on the scanning power lens, I want to move it to the middle of my field of view. So in other words, if what I was looking for was right there, okay, before I move on to the next magnification, I want to move it to the middle. Because if I don't, what's going to happen?
It's going to disappear when you increase the power.
Right. This next lens only sees that much area. So now the thing I was magnifying is outside the field of view. What's inside the field of view is greatly magnified, but I don't see as much of the slide as I did on the scanning power. So before I go up to the next power, I want to take that specimen and move it to the middle of my field of view. Okay, now here's how I do that. On the new microscopes, which are not good, okay, see? This is what happens when you cut money from education. You get this, ghetto microscopes. Okay, all right, so you'd have to actually use your fingers on the stage. This is called the stage. I'm never going to ask you to label a diagram of a microscope, by the way, okay? This is the stage. So I'm going to move the slide around with my fingers until I get that thing in the middle. That sucks. Hopefully you can use one of the old microscopes. They're better, okay? The old microscopes have these knobs here for the stage, and your slide will be inside the little silver part there that you can move around. It's way better because you don't put your fingers on here and push the slide down and make it go out of focus while you're trying to move the slide around. Okay, it's a pain in the neck to use those. So try and get one of the old ones. There should be enough because this is a small class. Okay, so you'll move it around until you get it on the end, or sorry, into the middle. Now I say the end because there's actually something in the microscope that makes it easy to find the middle. It's called a pointer. Okay, and the pointer
is going to look like a black spear. Kind of like this. Okay? And it indicates where the middle is. So put whatever it is you're looking at right on the end of that, and then it'll be in the middle. And then when you move up to the next magnification, you know it's still there. You might not see it because you got to get it in focus again, okay? But it'll be there. Everyone following me so far? Okay, then I'll get it focused, okay, on here. It'll look obviously much bigger, okay? When it's in here, it'll probably be, something like that now, all right? And then I would get it in the middle and go onto my high power lens. On the high power lens, I might only see that much of it, okay? It's greatly magnified, but the part of it I see is much smaller now because I've increased the magnification. Everyone follow the relationship there? Okay. All right, so that's what we'll have to do throughout the lab for each one of our specimens. Letter E, we're only looking at here. Amoeba, we're only looking at on low power because it'll kind of be like that. It'll take up pretty much the whole space. Okay, and then on high power, that's why we're not looking at the amoeba on high power. You won't see it all, but you will see all of the paramecium and all of the euglena on high power, which is why you should view them there. Now, each time you get it good and in focus on the proper magnification, you're going to take your phone, put the camera up to it takes a little while to get the hang of this, and get a picture of it with your phone through the microscope. Okay, you can do it. It just takes a little bit of practice, but you can do it. Okay, so you'll do that. Everybody with me there? Okay. Then we're going to make lab diagrams. Now, let's talk about how we do this. So in Google Docs, if I want to put my lab diagrams in, and it says here, see lab drawings on following pages, so I'm just going to go to the next page. Okay, and then I'm going to insert, if I get my toolbar right. There we go. Now I get the keyboard. Okay, I'm going to go to insert, and I'm going to go to drawing. Okay, we're going to use the worst graphics program ever made, Google Draw. Okay, but it's okay. All we need to do is take the picture we've taken with our camera, okay, and stick it in there and put some lines out to label it with. Okay, it's super easy. We're not actually doing any video or any photo editing or photoshopping or anything like that. Okay, so it'll work just fine for what we want to do. All right, and I'm going to go new. Okay, and then it opens up this dialog box. It's pretty simplistic. Right, so what I want to do then is I want to insert a picture that I already have. Okay, so choose an image to upload, and I've got an amoeba here all ready to go. Okay, so you would have to put your pictures in your Google Drive, obviously, for that part. Okay, so while it's uploading, okay, sweet, got my amoeba in there. Okay, now I want to be able to label it. Now there's a certain format that is acceptable for lab diagrams. Okay, and that is that your labels should all go to one side of the paper. Your diagram should not look like what you were looking at is exploding. Okay, because I often get lab diagrams where there are diagonal lines going everywhere, okay, out of the specimen. They should all go to one side and they should all be horizontal. Okay, so if I want to label a few things, I just click the line tool there. Okay, and I just start. Oh, of course it's not going to work on here because I'm on the touchpad. Okay, there we go. So I've got the line tool now, okay, and I'm going to draw a nice horizontal line off to the side. I'm going to label that thing, and I'm going to label that thing and probably that thing. And notice I'm making all my lines go out to about the same length. This one's a little bit short. And let's say I'm going to label that. Okay, so now I've got my labels there, okay, or my labeling lines, and I'm going to put text boxes on the end of each one of those. So that's this icon right here, okay, the text box, and again, I'm just going to draw that, okay, like so. Put a text box on the end of that, and just type in there cytoplasm, because that's what that is. Okay, and then I'll do the same thing, okay, for the vacuole. Okay, and any other things I need to label. Everybody with me on that? Okay, so that's how you do your labeling. For this lab, I don't care whether you label things correctly. I'm worried about the format of your diagram. So you could say clear bubble, stuff, big brown thing, okay? Well, I don't care what your labels are, okay? Not for the first one anyway, okay? On our next lab, I will care that you label everything correctly, but on this lab, I don't, okay? Especially when you're labeling the letter E, okay? Like you're gonna go like inside of E, ink, bottom of E, I don't know, like what do you label on the letter E? Pick a couple of things and just label them, call it whatever you want, okay? All right, so that's where your labels go, okay? And then format-wise, for a lab drawing, okay, your name and the date go in the top right corner. Okay, so you put a text box up in the top right corner, okay, and you put your name and the date up there. Don't write name and date. Put your name and the date. Okay. And then at the bottom of your diagram, this is the weird part, is where your title, size, and magnification go. All right. So you'll put again one text box down here at the bottom. Okay. And hopefully you have enough room. I didn't really plan my room out very well here. Okay. So I'll say amoeba, okay, and we're going to look at this on the low power lens, so the magnification will be 100 times, right? And then I have to estimate how big it is. Okay? To estimate how big it is, this is what I do. If I'm looking at something on, let's say, my scanning power lens. Let's say I'm looking at this little red thing on my scanning power lens. I know that the distance from one side of the circle to the other is 4,000 micrometers. So this thing here takes up maybe about a fifth of that, maybe a quarter generously. Okay, so it's probably somewhere between 800 and 1,000 micrometers. Okay, it's just an estimate. Okay, I'm not saying you have to measure and actually calculate how big this thing is. Okay, I look at that and go, I think 5 of those could fit across. That makes it about, so 4,000 divided by 5 is 800. Okay, done. It's 800. It's 800. Okay, so then I put in here. Okay, as my other line, 800 micrometers. Okay, you can write UM for micrometers. It's actually the Greek letter mu, which is the Greek letter M, because you can't go MM because that already stands for millimeters. Okay, and a millimeter is 1,000 times bigger than a micrometer. Okay, so if you write in there that your amoeba is 800 millimeters, you are telling me that your amoeba is that big. I don't think so. Okay? All right. Everybody follow on the lab drawing? So you're going to do one of those for each of your organisms. I have a separate recording of this exact procedure in with the lab. Okay, so if you're getting lost on it later when you're putting your lab report together, you can watch it again. Okay, but it's same deal. Okay, that's all you have to do. Okay, so now you can go, all right, save and close, and it inserts it right into your Google Doc. Okay, your lab diagram is done. It should take up an entire page. Okay, I don't want to have all 4 thumbnails. I can't tell anything on a thumbnail. All right, I'm old, so it should be a full page each. All right, so you can see there, okay, it takes up the majority of the page. It got cut off a little bit here, but everybody good with that? All right. Okay, I think that covers all of the stuff we have to know for Monday. Okay, so we're going to do those 4 things. You're going to take pictures with your phone. Okay, one member of each group can take the pictures and share them, or you can each try to take the pictures. Okay, we want to do this in partners. Okay, no bigger than that, because if it's bigger than that, then everybody doesn't get to use the microscope very much. All right, I would like each person to be finding half of the stuff you look at. So find a partner and work with a partner. Okay, for the lab. For the analysis part. Okay, so your lab diagrams are going to be your observations. Okay, this is a bit better job of a lab diagram. Okay, so I labeled a whole bunch of things. This thing and this thing here, they were too close together for me to make the lines horizontal. So I angled one and then went over as soon as I had room. Everything should end in a horizontal line, okay, on a lab diagram. Name and date in the top right, title, magnification, and size. Go underneath, okay, and then we should be good from there. Okay, that is the proper format for a lab diagram. Okay, for the analysis, okay, you got 3 questions there, okay, using what you saw under the microscope, and this is mostly going to be with your letter E, because that's the only one you know the orientation of. Okay, explain what you think happened to enlarge the specimens. That would be that diagram I drew the other day with the lens and the light and all of that. That's how a microscope works, so that's what you're saying there. Okay, for question number 2, I just want you to describe what you did to find and focus the paramecium. Okay, so kind of like a set of steps. So, you know, put it on scanning power, then I did this, and then I did this, and then I did this. Okay, and then for number 3, why do we add water to a wet mount? No, you cannot say it wouldn't be wet without it. I've had people say that. Okay, it's that whole polar thing. Okay, if we don't add water, okay, then how does the cover slip stay on? You need to kind of explain how the water keeps the cover slip on, okay? Because without it, it just blows away. Okay. All right, and then you got conclusion, same as always, copy paste your hypothesis. Did everything look the way you predicted? Does that tell you that the microscope works the way you thought? Right, and then sources of error. I'll give you one. Inexperience. There is nothing you can do about that. You are inexperienced using the microscope, and that is going to give you trouble. And there's an easy way to fix it. You got to come up with one more source of error. You'll figure it out once you get in the lab. Okay, trust me. All right, so that is what we will spend all of Monday's class on. Okay, we'll be in the lab using the microscope. And then we'll come back here. We won't have the Chromebooks, so there won't be, unless you're going to do it on your phone or something, there won't be electronics for you to use. All right, questions on that? Okay. So we've still got a few minutes here. 11:14. So you still got like 10 minutes. See how much of that pre-lab stuff, the variables explained, hypothesis done, you can get done. It's less to do later. Yeah.